Hello students. In our previous session, we had studied about the genetic code. We had understood what is genetic code. We had studied about the codons, the triplets of the nitrogen bases, which are called as codons. Each codon will be coding for one amino acid, right? And we had studied the salient features of the codons or the genetic code, and also we had looked into the checkerboard, which codon will code for which amino acid, and also we had studied about the mutations with respect to the genetic code, right? These mutations which are associated with a single nucleotide or a single nitrogen base are usually called as the point mutations, right? So under point mutations you have got frame shift mutations, maybe caused due to the addition or deletion, maybe substitution or maybe the deletion of nucleotide or one single nitrogen base. After understanding this, now see, genetic code is nothing but the language of what? It is the language which mediates from the language of the DNA to the language of the protein. While discussing about the genetic code, while talking about the genetic code, I had told you, see, these people, these different scientists like George Gamow or Khorana, right, Hargobind Khorana, all these people, many people, they tried only to decipher the code or the coded language that was carried from DNA to RNA, and this language itself is responsible for the production of the protein. But here, while copying the information from DNA to RNA, it is completely based on the rule of complementarity, right? But there is no complementarity rule. This rule of complementarity or the principle of complementarity will not work when the information has to get translated from mRNA to protein. During the synthesis of the protein, based on the information present on the mRNA, there will be no complementarity, right? But still, there will be specific amino acids brought together to form a polypeptide chain. How is this possible? This itself was answered by the concept of genetic code, right? They told that these four nitrogen bases, A, G, C and U, in the combination of three, that is in triplets, they will code for different amino acids. And in the checkerboard you have studied, there are 64 codons. Out of the 64, 3 are stop codons which will not code for any amino acid, and 61 codons will be coding for different amino acids. That we have already studied, right? So the codons or the sequence of the nitrogen bases present on the mRNA, that itself will be responsible for the synthesis of the protein, right? See, this much information we have already understood, and in today's session let us look into how exactly that information present on the mRNA, which is in the form of the genetic code, will get translated into protein. Translation or the synthesis of the protein, hege agutte actually, the process, how the process of the protein synthesis, mechanism of protein synthesis, which is also called as translation, how does it take place anta navu ivattu nodona. But before we go for looking into the details of the translation process, we need to understand one more very important thing, that is about the tRNA. See, you have already come across about different types of RNA. I have told you while learning about the transcription process. There are different types of RNA, mainly three different types. One is mRNA, and the other is rRNA, and the other is tRNA. mRNA stands for what? Messenger RNA. tRNA stands for transfer RNA. rRNA stands for ribosomal RNA. Here, this tRNA is also called as adapter molecule. It's also called as adapter molecule. Why it is called as adapter molecule? Here we are going to understand the details of the functionality of this tRNA, but before we go for that, let us look into the brief structure of the tRNA. Here the tRNA somewhat looks like this. Okay, so it somewhat looks like this. So to explain the model to explain the structure of the tRNA, a model has been proposed by a scientist, and that model is called as clover leaf model of tRNA. That clover leaf model will look like the leaf of the clove. It looks like this. So this model was proposed to give the or to explain the structure of the tRNA, right? And according to this structure, he says that there are 4 arms for the tRNA molecule. Here, this arm is called as amino acid acceptor end or acceptor arm. Arm or end both are same, or terminal anta kuda karibahudu, right? And opposite to that will be the anticodon end or anticodon arm, right? And this one was called as D arm, this one was called as T arm. Okay, so if you go to the detailed study, maybe in the NEET classes we can consider that each different arm will have the specific number of nucleotides there, right? So here also we can look into that. This is a structure which is normally drawn usually in your textbook also. This kind of diagram is given. Here this is nothing but what? Anticodon site or anticodon arm or anticodon end, and this one is what? It is the amino acid acceptor site or end or arm, right? Here you need to understand one thing. It is called as adapter molecule. Why is it called as adapter molecule, or why is it called as, it has been named as transfer RNA, is because it is going to transfer the specific amino acids. What? It's going to transfer the specific amino acids in the cell to the site of the protein synthesis in the cell, within the cell itself. The amino acids are also present in the cell itself, right? So those amino acids which are present somewhere in the cell, they have to be transferred, they have to be brought to the site of protein synthesis, right? That function is done by the tRNA. So that function of transferring the RNA molecules, transferring the amino acids cannot be done by any random tRNA. Here there will be always specificity in all these processes. All this mechanism will have very much specificity. Here anticodon site anta ಏನು ಕರಿತೇವೆ ನಾವು, anticodon arm ಅಂತ ಏನು ಕರಿತೇವೆ. Here whatever the 3 nitrogen bases are there, say for example CUG, if that is the sequence at this particular anticodon site anta andಕೊಳ್ಳಿ, then this will be complementary to the codon present on the mRNA. Try to understand. If this is the mRNA ಅಂತ imagine ಮಾಡ್ಕೊಳ್ಳಿ, 5 prime to 3 prime mRNA it is, and here this tRNA will come and bind to only that particular site which will be having a codon which is complementary to this one. ಇದಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಗಿರುವಂತಹ ಒಂದು ಕೋಡಾನ್ ಎಲ್ಲಿರುತ್ತೋ ಅಲ್ಲಿಗೆ ಬಂದು ಇದು tRNA ಸೇರ್ಕೊಳ್ಳುತ್ತೆ. ಹಾಗಾದ್ರೆ CUG ಇಲ್ಲಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ ಏನ್ ಬರ್ಬೋದು ಇಲ್ಲಿ? GAC. Yes or no? GAC. So GAC ಯಾವುದಕ್ಕೆ ಕೋಡ್ ಮಾಡ್ತಾ ಇದೆ? Which amino acid is coded by GAC ಅಂತ ನೋಡಿ ಸ್ವಲ್ಪ. GAC will be coded by aspargine. Sorry, GAC is going to code for aspargine. So GAC ಅನ್ನುವಂತಹ ಒಂದು ಕೋಡಾನು mRNA ಮೇಲೆ ಇರುವಂತಹ ಕೋಡಾನು GAC is coding for aspargine, ASP, aspargine or aspartic acid ಅಂತ ಹೇಳ್ತೇವೆ, right? So that means based on the anticodon arm, based on the sequence present in the anticodon arm, so if it is CUG, it will be complementary to GAC. ಹಾಗಾದ್ರೆ GAC will code for which amino acid? That is aspargine. This amino acid will be carried by this particular tRNA, aspargine. I hope you're understanding, right? So look at this one now. Another example let us consider. Here the mRNA is here, right? And I'm going to consider one simple codon here, that is AUG. You know, AUG is the start codon, correct? No? AUG not only acts as the start codon, it also has another function, that is it codes for methionine, right? So ಇಲ್ಲಿ AUG ಇದೆ ಅಂತ ಹೇಳಿದ್ರೆ ಇದಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಗಿರುವಂತಹ tRNA ಇಲ್ಲಿ ಬೈಂಡ್ ಆಗಬಹುದಾದಂತದ್ದು ಯಾವುದು ಅಂತ ಹೇಳಿದ್ರೆ ನೋಡಿ UAC. yes or no? That means the tRNA which has got the UAC sequence, ಆಂಟಿಕೋಡಾನ್ ಆರ್ಮ್ ಅಲ್ಲಿ UAC ಸೀಕ್ವೆನ್ಸ್ ಅನ್ನ ಹೊಂದಿರುವಂತಹ tRNA ಮಾತ್ರ ಎಲಿಜಿಬಲ್ ಆಗಿರುತ್ತೆ ಯಾವುದನ್ನು ಕ್ಯಾರಿ ಮಾಡಲಿಕ್ಕೆ, ಮೆಥಿಯೋನಿನ್ ಅನ್ನ ಕ್ಯಾರಿ ಮಾಡಲಿಕ್ಕೆ. ಮೆಥಿಯೋನಿನ್ ಅನ್ನುವಂತಹ ಅಮೈನೋ ಆಸಿಡ್ ಅನ್ನ ಕ್ಯಾರಿ ಮಾಡಲಿಕ್ಕೆ ಯಾವುದು ಎಲಿಜಿಬಲ್ ಆಗಿರುತ್ತೆ, ಸ್ಪೆಸಿಫಿಕಲಿ ಯಾವುದು ಅದು ಮೆಥಿಯೋನಿನ್ ಕ್ಯಾರಿ ಮಾಡಬಹುದು ಅಂತ ಹೇಳಿದ್ರೆ ದ tRNA which is having the anticodon sequence UAC. why UAC ಮೆಥಿಯೋನಿನ್ ಕೋಡ್ ಮಾಡೋದಿಲ್ಲ, ಅಗೇನ್ ಹೇಳ್ತಾ ಇದ್ದೀನಿ, ಕನ್ಫ್ಯೂಷನ್ ಮಾಡ್ಕೊಳ್ಬೇಡಿ. ನೋಡಿ ಇಲ್ಲಿ ಆಂಟಿಕೋಡನ್ ಆರ್ಮ್ ಅಲ್ಲಿ ಯಾವ ಸೀಕ್ವೆನ್ಸ್ ಇದೆ, ದಟ್ ಈಸ್ ನಾಟ್ ದ ಸೀಕ್ವೆನ್ಸ್ ವಿಚ್ ಇಸ್ ಕೋಡಿಂಗ್ ಫಾರ್ ದಟ್ ಆಸ್ಪರ್ಜಿನ್. ಸೀಕ್ವೆನ್ಸ್ ಯಾವುದು, ಇದಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಗಿ mRNA ಮೇಲೆ ಯಾವುದಿದೆ, ದಿಸ್ ಈಸ್ ದ ಕೋಡಾನ್. ದಿಸ್ ಸೀಕ್ವೆನ್ಸ್ ಇಸ್ ವಿಚ್ ಇಸ್ ಕಾಲ್ಡ್ as ಕೋಡಾನ್, ದಟ್ ಈಸ್ ಆಕ್ಚುಲಿ ಕೋಡಿಂಗ್ ಫಾರ್ ವಾಟ್, the amino acid. Here also AUG is coding for methionine, not UAC, right? So UAC ಅನ್ನ ಹೊಂದಿದ್ದು tRNA ಆಗಿದ್ರೆ that will go and bind to, that will go and fix to which one, the AUG. Then AUG is coding for methionine. That is why this tRNA will carry the particular methionine amino acid, right? So this functionality of the tRNA, that this particular type of RNA, that is why it is called as what, a transfer RNA or tRNA. Because of this its function, it's also called as adapter molecule. Adapter molecule ಅಂತ ಹೇಳಿದ್ರೆ adapter ಗೊತ್ತಲ್ವಾ ನಿಮಗೆ? So usually in the electrical shops you will get adapter. So it will go and it can be inserted into a particular plug, right? ಸೊ ಅಡಾಪ್ಟರ್ದು ಸೈಜು ಮತ್ತು ಪ್ಲಗ್ ಸೈಜ್ ಕರೆಕ್ಟ್ ಇದ್ರೆ ಮಾತ್ರ ಅಲ್ಲಿ ನೀವು ಅದನ್ನು ಕನೆಕ್ಟ್ ಮಾಡಬಹುದು, ಅದರ್ವೈಸ್ ಇಟ್ ಇಸ್ ನಾಟ್ ಪಾಸಿಬಲ್. ಹಿಯರ್ ಆಲ್ಸೋ ಇಟ್ ಇಸ್ ವೆರಿ ಮಚ್ ಸ್ಪೆಸಿಫಿಕ್. ದ ಆಂಟಿಕೋಡಾನ್ ಆರ್ಮ್ ಅಲ್ಲಿರುವಂತಹ ಸೀಕ್ವೆನ್ಸು ಮತ್ತು ಕೋಡಾನ್ ಸೀಕ್ವೆನ್ಸು ಅದೇನಿರಬೇಕು, ಸೇಮ್ ಅಲ್ಲ. ವಾಟ್ ಇಟ್ ಶುಡ್ ಬಿ, they should be complementary to each other. Then only this particular tRNA will bind to that, and then only ಕೋಡಾನ್ ಯಾವ್ದಿರುತ್ತೆ, ಆ ಕೋಡಾನ್ ಯಾವ್ದನ್ನ ಅಮೈನೋ ಆಸಿಡ್ ಅನ್ನು ಕೋಡ್ ಮಾಡ್ತಿರುತ್ತೆ, that amino acid will be carried by that particular tRNA. So that is why the transfer RNA or adapter molecule, the names have been given, right? So after understanding this, now we can easily understand by studying the process of the translation, we can understand the mechanism of the protein synthesis. Let me go for the next process, that is the protein synthesis, which is called as translation. Let us consider that now. See, translation is a common English term. Translate, ನೀವು ಕೇಳಿರ್ತೀರಿ, translation of a sentence from one language to another language. ನೀವು ಹಿಂದಿಯಲ್ಲಿ, ಸಂಸ್ಕೃತದಲ್ಲಿ ಅಥವಾ ಕನ್ನಡದಲ್ಲಿ, ಇಂಗ್ಲಿಷ್ನಲ್ಲಿ you already have learnt, right? So translating a sentence from one language to another language, right? That is what exactly is happening here. Translation of the information present in the form of or present in the language of the DNA and RNA into the language of amino acids or proteins. This process is called as translation. Okay, that means I am telling you this statement because you just need to realize why this process is called as translation. It is nothing but information which is present in the coded language or in the language of the nitrogen bases or in the form of the nucleotides, that will be converted into another language, the same information in another language of the amino acids and proteins. Yes, this process is called as what? Translation. Okay, so now let us look into the details of translation. How can you define translation? What is translation ಅಂತ ಕೇಳಿದ್ರೆ ಏನು ಹೇಳ್ಬೋದು? Translation refers to the process of polymerization of amino acids to produce a polypeptide chain, right? A process of polymerization of amino acids by peptide bonds to synthesize a polypeptide chain. To synthesize a polypeptide chain. So that is what is the definition of a translation. Right? Now, let us see how exactly this process is executed and what all the requirements are needed, what all the tools and techniques are involved in the process of translation. Where exactly is it taking place? That details we are going to study now. Of course, the translation is the process which involves all the different types of RNA molecules which are being synthesized during the process of what? The transcription. You know, transcription is the process which is responsible for the synthesis of different types of RNA molecules, mainly mRNA, tRNA, and also the ribosomal RNA, or also called as rRNA, right? These different types of RNA molecules, they work together, they work in coordination, and the site of the synthesis of the protein being the ribosome. Right? So this all happens within the cell, in the cytoplasm. Where exactly in the cytoplasm? In the ribosome. Right? So now this process of protein synthesis will also have the stepwise sequence. For our convenience, we can study the entire process in mainly 3 different steps. First one is initiation, then elongation, and termination. See, these processes are also the same processes which we have come across in the process of transcription, right? And while studying about the transcription, we had studied something called as transcription unit. Try to recollect. Transcription unit ant helidre enu? It is a sequence of DNA which is consisting of some structural genes which are flanked by the, which one? The promoter and the terminator. Right? There are 2 sequences on the DNA. One is called as the, which one? The template strand, and the other will be called as the coding strand. Right? So then similarly, here also we have got the mRNA. The mRNA will be there, and this mRNA having the polarity 5' to 3' will be consisting of what? It will be consisting of a start codon, and stop codon. Right? Start codon and stop codon will be there. Start codon towards the 5' end, then stop codon will be towards the 3' end, and here there are some untranslated region. UTR stands for untranslated region. Here also there are untranslated regions. You know the structure of the mature RNA, mature mRNA in eukaryotes, you know how it will be, right? It will be consisting of a methyl guanosine triphosphate cap, Mg 3-phosphate cap, and here towards the 3' end it will be consisting of a poly A tail. Correct? No? You know already methyl guanosine cap formation, which is called as capping, and it is called as tailing process. So a mature mRNA in the case of eukaryotes will be also consisting of the 5' untranslated region and 3' untranslated region, which are required for the efficient or smooth ongoing for the process of translation. If at all the process of the translation has to take place in a very efficient way or efficient manner, these have to be present on the mRNA, right? And in between the start codon and the stop codon, this is the start codon and this is the stop codon, right? You know which will be the start codon? It must be the sequence called AUG. Yes or no? And stop codon, there are 3 stop codons. Try to recollect from the checkerboard of the genetic code. There are UAG, UGA, and UAA. Right? Let us consider any one of them. UAG. Let me consider. That is the stop codon, which will not code for any amino acid. And in between them, illi AUG ante helidre definitely the methionine will come and join. Amino acid yav barbeku illi methionine barbeku, and similarly here any sequence, right, like C U G G CAUCAGUG. Okay, so these are the codons which are present in between the start and the stop codons, and these are also coding for different amino acids. Yes or no? See, this unit on the mRNA which is having the codons which are flanked by the start codon and the stop codon, this will be called as translational unit. This is called as translational unit. Of course, the diagram of this will not be asked, but you must know what is a translation unit. Transcription unit is different and translation unit is different, right? The diagram of transcription unit may be important for the exam, but this diagram won't be asked, don't worry. But this is just for your information. This is just for your knowledge. You must know what is translational unit. Only then you will be able to understand the process of translation. Right? See, you know what is translation process now? The definition you have understood, right? And of course, it takes place in 3 different steps: initiation, elongation, and termination, right? And it takes place where exactly? On the translation unit. Where is the translation unit? It is present on the mRNA, a mature mRNA, right? And so, but only mRNA. Is it possible for only the mRNA to carry out the process of the translation? Definitely not. It will require the participation of other participants or other molecules and other organelles also. Like, see, what happens here is, first thing, even before initiation, what happens is charging of tRNA is also called as amino acylation. Amino acylation of tRNA. This process will happen. This is the first and foremost step that we see in the process of translation. What happens here, and what exactly is the meaning of this one? Here, translation is the process which will be requiring a lot of energy. It's a high energy requiring process. This will require a lot of energy, right? And in order to supply that energy, for the supplement of that energy, the amino acids and tRNA, they will receive that energy in the form of ATP at the beginning of the process itself. And that energizing process by the usage of ATP molecules, where the amino acids or the tRNA molecules get energized, that process is called as charging of tRNA or amino acylation of tRNA, right? So why so much energy is required? You know the structure of the protein from the chapter of biomolecules in the first PU, that proteins are nothing but it's a polypeptide chain of amino acids. Many amino acids, they have been joined in a proper sequence with the help of which bond? Peptide bond. There will be peptide bond in between 2 successive amino acid molecules, and in order to form this peptide bond, there will be the requirement of high amount of energy, which will be supplied in the first and foremost step of this process, which is called as charging of tRNA or amino acylation of tRNA. Then what exactly is the meaning of this one? Is attachment of the particular the amino acid to a specific tRNA, right? Here, look at this. This is the tRNA molecule. This will be the anticodon arm, and this will be the amino acid acceptor arm or amino acid acceptor end anta kariteve, right? Now what happens here? This will be consisting of some anticodon. Say for example, any anticodon you name, like for example UCU. Of course, UCU, if it is the codon, then say for example here AGA. AGA is the anticodon sequence in the this will be carrying which one? Serine. Okay, See, UCU is the codon, then which will be coding for the serine. So ಹಾಗಾದ್ರೆ ಇದಕ್ಕೆ ಕಾಂಪ್ಲಿಮೆಂಟರಿ ಆಗಿರುವಂತಹ ಒಂದು ಆಂಟಿ ಕೋಡಾನ್ ಇತ್ತು ಅಂತ ಹೇಳಿದ್ರೆ, then that will be carrying the amino acid which can be coded by this codon. Right? Say for example another, that means, so based on the complementarity and based on which sequence is there in the anticodon arm, so that particular amino acid will come and bind to that particular, which one, tRNA. Right? Now another example let me consider. Right? See, this is another tRNA. Now say for example, let me consider any random sequence. Say for example UAC. UAC, that means the complementary of this one will be which one? AUG. Now can you tell me, AUG will code for which one? Methionine. That means this tRNA will be bound to methionine. It will be bound to the methionine. Amino acid methionine will bind to that particular tRNA. Right? So this is how the process of charging of tRNA or amino acylation of tRNA will take place in the presence of ATP. Right? So this process is called as amino acylation. Remember, it can be a very important one mark question. So what is amino acylation anta kelbodu, athwa what is charging of tRNA anta kelbodu, right? So this is the first step you have to write whenever you are explaining the process of what? The translation, right? Now after this process is done, now what happens? Let us see what happens next here. Initiation. In the process of initiation, see, already the tRNA molecules are ready with one particular amino acid. Yes or no? Say for example, illondu amino acid, illondu tRNA ide, and this is carrying already one amino acid. Say for example, methionine anta andkolli. Now this is, what is this? mRNA sequence. mRNA. Okay, this is the mRNA, right? And say for example, if this is the UTR, this will not code for anything, untranslated region anta irutte idu, and from here it will be having AUG anta andkolli, right? AUG ಅಂತ ಅಂದ್ಕೊಳ್ಳಿ. Now what happens? So methionine carrying tRNA is already there with the which one? UAC anticodon, right? So ಇದು ಇಲ್ಲಿಗೆ ಬಂದು ಸೇರಬೇಕು. But will that happen all of a sudden? That means ಇಲ್ಲಿ mRNA tRNA on its own, will it happen like that? Definitely not. It will require an assembly of the ribosome. It will require the catalytic site called as ribosome. So ribosome site hegirutte, you know, the ribosome will be consisting of two subunits. The smaller subunit and the larger subunit. So this is the larger subunit, this is the smaller subunit. You know, 80S ribosomes, 70S ribosomes, which are made up of two different types of subunits, right? This larger and smaller. Now first ಏನಾಗುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ, the mRNA will come and bind to the smaller subunit of the ribosome. I'm repeating, the mRNA will come and bind to the smaller subunit of the ribosome. Where? In the process of initiation I'm talking. In the process of initiation, the mRNA will come and bind to the smaller subunit of the ribosome. Later, the larger subunit also will come and bind, and only then it will become a complete unit or the complete site, which will be a functional and catalytic site for the synthesis of the proteins, right? Both of the subunits have to be there. Now, this ribosome will act as the catalytic molecule and it will provide three main sites. Andre nodi, there will be some sequence here. Say for example, AUG illide anta andkolli. Okay, imagine AUG is here. So AUG is the start codon. So this cannot be here. AUG ille irbekalva. AUG start codon alva, that has to be here, and it will be followed by many amino acids. Say for example, GUC, CUA, right? AUC, then UCA, then ACU, right? And last one should be which one? UAA, because stop codon irbeku alva. Now here ಇಲ್ಲಿ ಏನಾಗಿರುತ್ತೆ ಅಂತ ಹೇಳಿದ್ರೆ imaginary 3 sites will be there. Like for example, see this one, which one? AUG, GUC, CUA, and here let me consider AU and C. AUC. See, this is the site. This is one site having one codon, and this site is called as P site. Okay, and here CUA. Here is another site. This site is called as, this is called E site, and there is one more here. This site is called as A site. Okay, that means initiation ಆಗುವಾಗ ಏನಾಗುತ್ತೆ, ಫಸ್ಟ್ ನಾನು ಹೇಳಿದ್ದು ribosome, smaller subunit of the ribosome will come and bind to the which one? The mRNA and smaller subunit will come and bind, and then the larger subunit of the ribosome will also come and bind, forming the complete working unit of the ribosome for the process of protein synthesis. Now this catalytic unit will provide 3 main sites. They are called as E, P and A. E stands for exit site. P stands for peptidyl site. E stands for exit site, and A stands for acceptor site. ಏನಿದು ಹೀಗಂದ್ರೆ, see here always AUG is the start codon ಅಲ್ವಾ, ಸೊ ಅದು ಯಾವಾಗಲೂ ಇಲ್ಲಿ ಬಂದಿರುತ್ತೆ, ಬಟ್ ಯಾವಾಗ ಸ್ಟಾರ್ಟಿಂಗ್ ಅಲ್ಲಿ, ಸೊ P ಸೈಟ್ ಅಲ್ಲಿ AUG ಇರುತ್ತೆ ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ಸೊ AUG ಇದ್ದಾಗ ಏನ್ ಬರಬೇಕು, ಮೆಥಿಯೋನಿನ್ ಅನ್ನ ಕ್ಯಾರಿ ಮಾಡ್ತಾ ಒಂದು tRNA ಬಂತು, AUG ಇತ್ತು ಅಂತ ಇಮ್ಯಾಜಿನ್ ಮಾಡ್ಕೊಳ್ಳಿ. ನಾವು AUG ಇದ್ದಲ್ಲಿ ಇಲ್ಲಿ ಒಂದು ಬಂತು. Acceptor site will be open like UCA. Let us see, UCA will code for which one? UCA, it is going to code for, UCA codes for ಸೆರಿನ್. UCA ಕೋಡ್ಸ್ ಫಾರ್ ಸೆರಿನ್ ಅಂತ ಹೇಳಿದಾಗ ಸೆರಿನ್ ಅನ್ನ ಕ್ಯಾರಿ ಮಾಡ್ತಾ ಇನ್ನೊಂದು ಇಲ್ಲಿ ಬಂತು ಅಂತ ಅನ್ಕೊಳ್ಳಿ. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ಪೆಪ್ಟಿಡಿಲ್ ಸೈಟ್ ಅಲ್ಲಿ ಇರುವಂತಹ ಒಂದು tRNA ಏನಿದೆ ಮತ್ತು ಇಲ್ಲಿ ಆಕ್ಸೆಪ್ಟರ್ ಸೈಟ್ ಅಲ್ಲಿ ಇನ್ನೊಂದು tRNA ಇದೆ. ಎರಡೂ tRNAಗಳು ಒಂದೊಂದು ಅಮೈನೋ ಆಸಿಡ್ ಗಳನ್ನ ಕ್ಯಾರಿ ಮಾಡ್ತಾ ಇವೆ. ಸೊ ದಿಸ್ ಇಸ್ ದ ಸೈಟ್ ವೇರ್ ದ ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್ ಫಾರ್ಮೇಶನ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್. ಐ ವಿಲ್ ಟೆಲ್ ಯು ಹೌ ಇಟ್ ಇಸ್. ಸಿ ನೋಡಿ, UAC ಇಲ್ಲಿದೆ, ಆಲ್ರೆಡಿ ಇದು ಬಂದ್ಬಿಟ್ಟಿದೆ, ರೈಟ್? ಸೊ ನಾವು ಅಮೈನೋ ಆಸಿಡ್ ಇಲ್ಲಿ ಫಸ್ಟ್ ಮೆಥಿಯೋನಿನ್, ಇನ್ನೊಂದು ಯಾವುದೋ ಬಂತು ಸೆರಿನ್, ಗ್ಲುಟಾಮಿನ್. ಓಕೆ, ಐ ಆಮ್ ರೈಟಿಂಗ್ ರ್ಯಾಪಿಡ್ಲಿ. ದೆನ್ ಫಿನೈಲ್ಅಲನಿನ್, ಐಸೋಲ್ಯೂಸಿನ್. ಓಕೆ, ಸಮ್ ಸೀಕ್ವೆನ್ಸ್ ಆಫ್ ದ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಆಲ್ರೆಡಿ ಹ್ಯಾಸ್ ಬಿನ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್, ಯಾಕಂದ್ರೆ ಇಲ್ಲಿಂದ ಶುರುವಾಗಿದೆ ಅದು ಅಲ್ವಾ. ದೆನ್ ಫಿನೈಲ್ಅಲನಿನ್ once again. Now AUC will code for which one? Let us see. AUC is going to code for isoleucine. AUC isoleucine ಕೋಡ್ ಮಾಡ್ತಾ ಇದೆ, ಬಿಕಾಸ್ ದಿಸ್ ಇಸ್ ದ ಕೋಡಾನ್. ಸೊ ಹಾಗಾದ್ರೆ ಇಲ್ಲಿ ಒಂದು ಅಮೈನೋ ಆಸಿಡ್ ಬಂದಿದೆ, ಒಂದು tRNA ಬಂದಿದೆ, and this tRNA is carrying which one? It is carrying ಐಸೋಲ್ಯೂಸಿನ್. ಹಾಗಾದ್ರೆ ಐಸೋಲ್ಯೂಸಿನ್ ಕ್ಯಾರಿ ಮಾಡ್ತಿರುವಂತಹ tRNA ಅದು ಆಂಟಿಕೋಡನ್ ಏನಿರಬೇಕು? UAG. UAG is the anticodon. So the tRNA with the anticodon UAG is carrying the isoleucine amino acid, and that has come here, right? But ಇಲ್ಲಿ ಹೇಗೆ ಬಂತು ಅಂತ ಹೇಳಿದ್ರೆ, ಫಸ್ಟ್ ಡೈರೆಕ್ಟ್ ಇಲ್ಲಿಗೆ ಬರಲಿಲ್ಲ ಅದು. ನೋಡಿ, ಈ ರೈಬೋಸೋಮ್ ಹೇಗಿದೆ ಅಂತ ಹೇಳಿದ್ರೆ, ಫಸ್ಟ್ ಇಲ್ಲಿತ್ತು ದ ಎಂಟೈರ್ ರೈಬೋಸೋಮ್. ನೀವು ಟೈಪ್ ರೈಟರ್ ಮಷಿನ್ ನೋಡಿರ್ಬೋದು. ಸಿ, ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಆನ್ ಮೂವಿಂಗ್, ರೈಟ್? ನೌ ಹಿಯರ್ ಸಿ, UCA will code for serine. Now this tRNA will be consisting of AGU. as the anticodon, and that is why it is going to carry the serine as the amino acid, right? So ಇಲ್ಲಿ serine ಬಂತು ಅಂತ ಹೇಳಿದ್ರೆ ಇಲ್ಲಿ ಆಲ್ರೆಡಿ ಇಲ್ಲಿ ಐಸೋಲೇಷನ್ ಇದೆ. ಈಗ ಐಸೋಲೇಷನ್ ಏನ್ ಮಾಡುತ್ತೆ, ಇಲ್ಲಿ ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ಕೊಳ್ಳುತ್ತೆ, ರೈಟ್? ಆಲ್ರೆಡಿ ಒಂದು ಪೆಪ್ಟೈಡ್ ಚೈನ್ ಇದೆ, ಇಲ್ಲಿ ಮೊದಲೇ ಬಂದು ಸೇರ್ಕೊಂಡಿರುವಂತ ಅಮೈನೋ ಆಸಿಡ್ಸ್ ಇದೆ. ಸೊ ದಿಸ್ ಅಮೈನೋ ಆಸಿಡ್ ಆಲ್ಸೋ ವಿಲ್ ಗೋ ಇನ್ ಫ್ರಮ್ ದ ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್ ವಿತ್ ದಿಸ್ ಒನ್, ಅಂಡ್ ದೆನ್ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್, ಈಗ ಇದ್ರ ಜೊತೆ ಐಸೋಲೇಷನ್ ಹೋಗ್ಬಿಡ್ತು. ನೌ ಐಸೋಲೇಷನ್ went and joined with the chain. Now what will happen here? See, this will move here. ಈಗ entire assembly of the ribosome next codon ಗೆ ಹೋಯ್ತು. ಆಗ CUA ಇಲ್ಲಿಗೆ ಬರುತ್ತೆ. ಈಗ AUC ಇಲ್ಲಿಗೆ ಬಂತು, AUC ಇಲ್ಲಿಗೆ ಬಂತು, UCA ಇಲ್ಲಿಗೆ ಬಂತು, ACU ಇಲ್ಲಿಗೆ ಬಂತು, UAA ಇನ್ನೂ ಹತ್ರ ಬಂತು ಅಲ್ವಾ? Just that means the entire ribosome assembly moved to the next codon. ಹಾಗಾದ್ರೆ ಇಲ್ಲಿ ಐಸೋಲೇಷನ್ ಅನ್ನು ಕ್ಯಾರಿ ಮಾಡಿರುವಂತಹ ಯಾವುದು tRNA ಇತ್ತು. So that already isolation has been transferred, and now what is remaining? Only the tRNA. Only tRNA. So this tRNA will get exited. It will get exited. ಅದಕ್ಕೆ ನಾವು ಎಕ್ಸಿಟ್ ಸೈಟ್ ಅಂತ ಕರಿತೇವೆ, ರೈಟ್? ಇಲ್ಲಿ ಒಂದ್ಸಲ ಅಮೈನೋ ಆಸಿಡ್ ಬಂದು ಪೆಪ್ಟೈಡ್ ಬಾಂಡ್ ಫಾರ್ಮೇಷನ್ ಮಾಡ್ಕೊಳ್ತು ಅಂತ ಹೇಳಿದ್ರೆ ದ ಓನ್ಲಿ ಎಂಪ್ಟಿ tRNA which is not carrying any amino acid will move to the this one, and then it will get exited, right? But here ಇಲ್ಲಿಗೆ ಯಾವ್ದು ಬಂತು? UCA. That means it is carrying which one? The serine. So now it is carrying the serine. So serine ಅನ್ನು ಕ್ಯಾರಿ ಮಾಡ್ತಿರುವಂತದ್ದು ಅಮೈನೋ tRNA ಇಲ್ಲಿಗೆ ಬಂದಿದೆ, and this serine amino acid will also forms bond with the peptide bond with the already existing amino acids, and once again when this happens, peptide bond formation ಆದ ತಕ್ಷಣ ಏನಾಗುತ್ತೆ? See, it will move. ಇಲ್ಲಿ ACU ಇದೆ ಅಲ್ವಾ? ACU ಅಂತ ಹೇಳಿದ್ರೆ that ACU will code for which amino acid? Let us say ACU is going to code for thrombin. ಆಗ ಈಗ ಸೆರಿನ್ ಅನ್ನು ಇಲ್ಲಿಗೆ ಬಿಟ್ಟು ದಿಸ್ tRNA will move here. ಆಲ್ರೆಡಿ ಇದು ಮೂವ್ ಆಗಿದೆ. ಇದು UCA ಇಲ್ಲಿಗೆ ಬರುತ್ತೆ, ACU ಇಲ್ಲಿಗೆ ಬರುತ್ತೆ, UAA ಇಲ್ಲಿಗೆ ಬರುತ್ತೆ. UAA when it comes to the acceptor site, it is not coding for any amino acid. That means the process of the translation will stop. Right? So initiation ಏನಾಗುತ್ತೆ? The ribosomal complex will be formed, right? And then initiation will be carried out by initiation factors. Elongation ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ? So the chain elongates. ಒಂದರ ನಂತರ ಇನ್ನೊಂದು, ಮತ್ತೊಂದರ ನಂತರ ಮತ್ತೊಂದು, like this. So based on the sequence of the amino acids, so a long chain of peptide bond or a peptide chain or the amino acid chain will be formed in the elongation with the help of elongation factors. Right? And termination will happen only when the termination sequence or the terminator codon UAA, UGA or UAG ಮೂರರಲ್ಲಿ ಒಂದು ಬಂದಾಗ which will not be coding for any amino acid, then that is when this process will be stopped, and release of the amino acid chain will be happening, and that will be mediated by terminator, what is that? Factor. Termination factor or termination factor, TF ಅಂತ ಕರಿತೇವೆ.